Okay. So, now we're going to talk about the replication machinery for our lovely DNA. As I mentioned before, and this is important and it bears repeating, the DNA that we have gets split into two strands, and I'm drawing this very intentionally, the way that I'm drawing it, into two strands, and then it gets replicated. So, and then it's kind of replicated out here with a friend. And now we're going to go through the ways in which our DNA gets replicated and all of the machinery required. So, helicase. So, DNA is coiled up. As we mentioned before, your nitrogenous bases are hydrogen bonded together. They like to stick together in the pairs that we told before. If you've got a mnemonic for that, you should be able to remember which pairs go together. And they stick to each other. And that means that it's hard to separate the DNA. And so in order to separate the DNA, we have a friend, and that friend is called helicase. And what helicase does is it's just a ring that sits around the DNA and kind of forces them apart. Very straightforward. Then, and I should probably, And then, as we uncoil the DNA, it kind of upstream, it gets more wound up. It can also just be wound up wrong in general, just because it's a really, really, really long string, and if you put your headphones in your pocket, then they're going to get coiled up. And so we need a way of uncoiling it, and what does that is topoisomerase. And topoisomerase, if I can draw it really quickly, it takes your DNA, breaks it, and puts it around the other side. Something like that. And it's got fingers and stuff to grab it, and it's kind of... I can go through that later. Topoisomerase twists around your DNA and stops it from getting super duper coiled by your helicase, which is separating the stuff into two strands. Okay, now single-stranded binding proteins. I haven't drawn those, but single-stranded binding proteins, and I'm going to go through all of this again at the end, but we're just going to get it all. Single-stranded binding proteins keep the DNA from clicking back together before we can replicate the DNA. So here and here, before it's been duplicated, we've got single-stranded DNA. And that single-stranded DNA is looking for friends. And it wants to hydrogen bond with whatever it can find a hydrogen bond. So if you've got stray nucleic acids, or over here you've kind of got a group, a sequence that can pair with that sequence, they would attach, and that would be bad, because then our downstream components really wouldn't work. And so that's what single-stranded binding proteins do, right? Single-strand binding, relatively straightforward. It binds to a single strand. SSB binds to a single strand. Topoisomerase, I can get that it's kind of harder to remember, but it's topo, which, topologies. So it sorts out the topology of the DNA. Topoisomer, it's a specific configuration of a polymer, and Ase is a what's the word? It's a protein that does things. And so helicase, same deal. It unwinds the helix. It unwinds the double helix. And okay, primase. So in order to understand the importance of primase, we need to talk about polymerase. DNA polymerase. In particular, we need to talk about polymerases 1 and 3. So I'm going to erase this stuff. I'm going to go down to our little diagram, and we're going to get into it. Okay, so might make a new video, and then I'll talk about the leading and lagging strands.